Hello, I'm Seleem Badawy, and today I will be telling you about the research that I have been conducting for the past 2 years in the Rinker lab, investigating fluid shear stress effects on postpartum breast cancer progression. Breast cancer is one of the most significant types of cancer. With 2.26 million new cases globally in 2020, we're seeing a significant effect of breast cancer on particularly young women. What these two graphs are telling us is that while breast cancer for older women has leveled off in terms of mortality, for younger women, it's actually stopped decreasing as a mortality rate, and it's actually begun to reverse. One of the key things to know for that as well is that mammography, which is considered the gold standard for identifying and diagnosing breast cancer, is actually not suitable for young women's breast cancer. Postpartum breast cancer is a type of breast cancer that affects women, particularly young mothers, within 5 years of childbirth. So these are new mothers and very young women. It is not well understood relatively to other types of breast cancer, and it lacks screening and diagnostics, causing it to often be diagnosed with not only stage 4 developed diagnoses, but also typically existing spread of the cancer throughout the body. So this is very significant because it causes an increased risk of death and distant spread of the cancer for this vulnerable subsection of women. After giving birth, the mammary gland goes through a few distinct stages. So first it goes through lactation, where it produces the milk, and then cessation, where it halts the milk production, and finally involution, where it returns the mammary gland and the breast to its regular pre-pregnancy state. This involution stage activates a lot of different mechanical processes because it's effectively returning the breast back, and so what it's doing is it has a high amount of cell overturn, and it has a lot of different processes such as wound healing and inflammation that cause a very high mechanically stressful environment. And so this stage in particular is hypothesized to contribute to breast cancer progression. So to investigate this, our objective was to understand the effect of fluid shear stress on postpartum breast cancer progression with the long-term goal of ultimately developing effective diagnostics. So what we wanted to do is approach it from an engineering approach. So we used a bioreactor-based model, which I will describe in a second, to better mimic the cell environment. Next, we had a very solution-oriented approach where we wanted to specifically have and identify biomarkers and other possible ways so that we could diagnose this cancer sooner. And finally, we had a particular focus on looking at the mechanical stresses on these cells. This is our bioreactor-based model that we've developed in the Rinker lab. Our aim was to study both the effect of fluid shear stress as well as the effect of hormonal influences on the cells. So we wanted to study first cells with just fluid shear stress. So that's what you see in this pathway to the right here. So these are cells, and these are just put with the media, they're untreated, and the regular cells will then just go through the flow chamber, which the flow chamber will essentially, in this chamber below here and with a constant media flow on top of it, exert a constant fluid shear on the cells that we could control, and we have set amounts of shear that are at each stage that we wanted to investigate. The other pathway, which is to the far left here, is the hormone-treated one. So this one is where we actually add hormones to the cells in 3 distinct stages in the amounts that would be found in the breast so that we could have the effect of the hormones present, and then we can run it through the fluid shear stress. After this, the samples are taken from the flow chamber and then studied. Now, some of our results that we have found so far is first that CSN2, which is a milk protein called beta-casein, has higher results in the hormone-treated versus untreated cells. What this indicates to us is that it validates our bioreactor model in terms of the hormone treatments because CSN2 is a lactation protein that would be expressed once lactation begins. So we could see that in our hormone treatments, they are effective in actually beginning this process. The next key thing that we've found so far is that tumor-related protein levels, such as CXCL13 in the Western blot graph to the left, which essentially just looks at protein levels in each of our different samples, has higher levels in the flow versus static, so the fluid shear stress affected versus the non-fluid shear stress affected cells, hormone treated versus untreated, so the hormone treated cells versus the untreated cells, and then flow 1 stage, which is the lactation, versus flow 2, which is cessation. This indicates a few things. Firstly, that it does suggest a role of fluid shear stress as well as of hormones on the actual levels of this protein. And so this protein in particular is involved in the initiation of tumors and the actual progression and spread of tumors, so progression of cancer overall. So in conclusion, we've found that our bioreactor-based model is an effective model to study cells under hormones and shear stress, which is important because now we can mimic the cell environment and effectively study these cells that would be in postpartum breast cancer. We also found that we have a suggested impact of fluid shear stress and hormonal influences on postpartum breast cancer, which opens a whole new doorway for our next steps where we want to continue studying both the proteins as well as the genes that are in these different conditions, and hopefully find something that can ultimately develop a diagnostic for this cancer. I would like to acknowledge the whole Rinker Lab, in particular Dr. Christina Rinker and Maya for being such incredible mentors, and all of our supporters. Thank you so much.